So I want to thank everyone for coming to the first Andlinger Center for Energy and the Environment Highlight Series seminar, and it's a real pleasure for 2014. Did I say 2013? Okay, good. Okay, for the first one of 2014. It's a real pleasure to have Professor Bruce Logan from Penn State University, who on some very heroic efforts on his part made it for the seminar. So we're really thrilled to have him. Dr. Logan is an Evan Hugh Professor and the Stan and Flora Kapp Professor of Environmental Engineering and the Director of the Engineering and Environmental Institute at Penn State University. His main research efforts are in bioenergy production and the development of an energy-sustainable water infrastructure, something that we've heard a bit about in the previous semester, so it's thematically coherent and a fantastic addition to what we've been really thinking about here. He is the co-author of over 350 refereed publications and a member of the National Academy of Engineering and a fellow of the AAAS. So without further ado, Dr. Logan, thanks so much for being here.
Yep.
Oh, and so please silence your cell phones and remember during questions to wait for the microphone as this lecture is being recorded. Thanks so much.
I forgot to do my own. Well, thanks very much for that nice introduction. Going to really talk about a few different things today, but mostly it's all about the water-energy nexus. And so as an environmental engineer, I spent a lot of time thinking about water treatment, sanitation, and these sorts of issues. And as you look globally, the situation's not getting better. It's not good right now. 2 billion people lack adequate sanitation. And by 2025, they say a quarter of people could be in severe water shortage areas, and by 2050, I think it could be like half the world. So you start thinking about water, and then you say, well, water isn't a problem if you have enough energy. But of course, if you use a lot of fossil-derived energy, then you have a problem of contributing to climate change. And the more you can contribute to climate change, the more that changes water patterns and water distribution, and the more energy you have to put into your water infrastructure, and it just starts to go in a circle and spin faster. So that really made me come back and start thinking a lot more about energy, and I want to put energy in two different perspectives. So first, the big perspective, the global use, 13.5 terawatts, and in the US, we use about 3.3 terawatts, maybe 600 gigawatts of electrical power used. And again, big picture, a typical nuclear power plant is about 1 gigawatt. Okay, so that's the bigger picture size. What about at a smaller scale? That would be you. So if you were to think about the food you eat every day, that's like 2,000 kilocalories, or 2,000 calories, or where capital C calorie is a kilocalorie, or about 8.4 megajoules per day. I don't know if those numbers mean anything to you, but you think about that and you say, well, I'm pretty sure there's a lot of energy in food, because every day I eat probably too much food and I get fat. So it must be, it must be a lot. So. But if you think on energy per day units and you equate that to how many 100-watt light bulbs would be running continuously, how would you guess that that's 1, 10, 100, 1,000? What's the equivalent? The number isn't probably readily at your fingertips because of the different units, but it's one. One 100-watt light bulb like this is the equivalent energy per day of the food you eat. And that should make you rethink about how much energy you're really using, because we use about 100 times that per person. Now, we're not perfect. The wastewater that is collected from our homes and that goes to the treatment plant is equal to about 25 watts per person. So we're pretty efficient, though. That's not bad. Turns out that's equal to about a compact fluorescent that puts out the same amount of light as this old technology there. So the energy perspective from the water infrastructure is that we use about 30 gigawatts of electrical power for our water infrastructure in the US, and about half of that has been estimated to be used for wastewater treatment. And I'm not going to try and defend or say those numbers are accurate, but they give you a fair idea about the kinds of power requirements we're talking about. For wastewater treatment, for drinking water treatment, the numbers are pretty small, but if you go to something like desalination, which we're increasingly doing to supply water, then the energy requirements start to come up on the order of 4 to several hundred kilowatt hours for a cubic meter of water. So I thought about all this and I said, well then, this is pretty simple. I want to make the water infrastructure energy sustainable, because if we can do that, we'll have enough water and we won't need to worry about the energy, because otherwise there's no way that the globe can have good water treatment and sanitation with these sorts of energy requirements. So, next question, how do I achieve this goal? And there was a very simple answer. Just develop a sustainable, green, CO2 neutral method of renewable energy production. So I thought, well, where am I going to get the water from? Well, the only thing I really understand is water, so I might as well get it from the water. Turns out that the energy we use for wastewater treatment, that's that 15 gigawatt number, if you look at how much energy is in just a few major sources of wastewater, the organic matter in there is equal to about 17 gigawatts. So there's as much energy in that wastewater as we need to run the water infrastructure. As we need for wastewater treatment. So if you work that down to cubic meters, it's about a half a kilowatt hour per cubic meter of energy used, but in just regular domestic wastewater, there's about 2 to 5 kilowatt hours per cubic meter. So there's much more energy in domestic wastewater than we currently use to treat it. So why don't we try and extract that energy? And if we can succeed at that, we could start to use those technologies to do other things. So for example, there's a lot of cellulosic energy out there. We don't want to burn the cellulose, so how do we make efficient use of it? Well, we could break it down, get it in water, and use the same technologies to generate energy from cellulose. And now, we're not talking about 17 gigawatts, we're looking at 600 gigawatts, maybe from just waste biomass. There's also a couple other good sources of energy that we could tap into if we could figure out how to do it. I know Manny Elamelik was here from Yale and talked about salinity gradient energy. There's maybe 2 terawatts available, and about maybe half of that could be reasonably harvested to provide energy. And waste heat energy is another great source of energy, because there's probably, again, a terawatt out there of waste heat just from the power plants that we're using, because a power plant puts 1/3 of that power into the electricity, and 2/3 of that just is wasted as heat. So this led me to the technology we call microbial fuel cells, and this is a microbial fuel cell. The bacteria break down the organic matter. They generate an electrical current. That current can be harnessed to do useful work, here shown running a fan. So this is kind of a crazy idea. It's kind of a radical idea. We want to develop a new technology based on microbial fuel cells for wastewater treatment. Well, new ideas always go through 3 stages. The first one is, it can't be done. I tell you that is very true for me, because my first NSF proposal was rejected. I said I wanted to turn a wastewater treatment plant into a power plant. They went, we know our motto is, we like high risk, high reward research, but we don't mean that risky. The second stage is, it's possible, but it's not worth doing. It's too expensive. And then the third stage is, I said it was a good idea all along. So there are examples of that in wastewater treatment, reverse osmosis membranes for water desalination. They said it'd never work. Membrane bioreactors for wastewater treatment. Stupidest thing in the world would be to put a membrane in a wastewater treatment tank, and these have certainly become mature technology. So maybe microbial fuel cells is the next one. So let's dissect this a little bit. In the broad category of microbial electrochemical technologies, or MFCs, where that little letter in the middle changes, here are the components. You have an anode and a cathode, 2 electrodes. The anode may have microbes on it, no, it's a bioanode. The cathode may or may not have microbes on it. One of these electrodes, if they have microbes on it, makes it a microbial technology, or both can have microbes. Then you can either make electrical power, or by adding in some electrical power, you can do some other things. And in some of these systems, there are no membranes. In others, there's one or more membranes in between the electrodes. So in the simplest microbial fuel cell, there's no membranes. The microbes grow on the anode. They break down organic matter. That's the fuel. They release protons into the water, electrons into the electrode and circuit. They flow to the counter electrode. There's an electrochemical potential difference, so it's spontaneous. It generates power. And all you really need to do this is take 2 pieces of carbon cloth, stick them, one exposed to air and water on the other side, and then one kept away from the air, and you can make electricity. So it's a very simple idea. If you take that same system and now you take away that oxygen from the cathode, so there's no oxygen here, absolutely nothing happens because there's not enough voltage produced to do anything at this electrode. But if you boost that voltage just a little bit by adding an external power source, then you can actually drive hydrogen evolution because you got 2 electrons going here, 2 protons here, 2 electrons, 2 protons, and you make hydrogen. So like a water electrolyzer, but here you're using that organic matter to supply a lot of that energy, and so you can do that at a voltage that's about 1/10 of what it takes to split water. If you further modify that, you could make other fuels. Some people say, well, I don't like hydrogen. Well, first of all, if you have organic matter, you can just make some hydrogen, but electrochemically, if you take that anode and make it abiotically, and you use some renewable source of power, you can drive an electrical current through here, and then you can either evolve hydrogen, or if you put methanogens on these electrodes, you can generate methane from that hydrogen. And at the same time, you're fixing CO2 into that methane. The nice thing about methane, of course, is that it would fit in well with our current infrastructure, natural gas, and increasingly anaerobic digesters at wastewater treatment plants. So it'd be a nice way to generate a transportable fuel.
Excuse me, by methanogens, you mean enzymes?
I mean microorganisms that fix carbon and make methane.
Okay.
So it's a whole cell.
Oh, the whole cell.
The whole cell.
So a bacteria?
No. Well, technically, it's not a bacterium, it's an archaeum. It's an archaea, it's a different tree of life.
Okay.
Yeah, but a microorganism. Self-replicating and so forth. So this is the basic technology. You can surround it with all sorts of different things at the anode. Typically, I'm just going to talk about feeding it acetate or a very complex source of organic matter. At the cathode, you can have some of these reactions that will produce power, oxygen, nitrate, and so forth, or you can use other microbes that will transform things like hydrogen that's evolved, but you need to add generally a little bit of power into the system to make it do that. So that's a little bit long introduction, and I'm going to now quickly jump into about 3 different areas and conclude 4th just very, very briefly. So the first one is I want to talk about these microbes. Second, I want to talk about these technologies and the materials and the potential for being real technologies. And then the third, I want to talk about salinity gradient energy, and I'm going to talk very, very little about what Professor Elmelik talked about and talk about some other technologies. So first, the electromicrobiology, which we consider sort of a new subfield of microbiology. There's a very modern laboratory that you would mistake as being an electrical engineering laboratory, but this is what now electromicrobiology laboratories look like. There are more potentiostats, shall we say, than petri dishes in this system. We can talk about electrogens, the microbes that release electrons to the anode, or electrotrophs, microbes that accept electrons from the cathode. So these are sending the electrons around, and these are eating electrons, because in nature likes cycles. So if something's making electrons and releasing electrons, it stands to reason that there are other microbes out there which can accept these electrons. Let's talk about the electrogens first. You take a microbial fuel cell, you throw in any source of water that has bacteria in it, the bacteria start growing on that electrode, form colonies, and they can transfer electrons directly to an electrode by some different mechanisms. The most fascinating is that they can make their own nanowires to conduct those electrons from inside the cell to outside the cell. Now, we've, as a society, been getting wired for a couple hundred years, but these microbes have been doing it for maybe 1 billion years. They not only can make these nanowires, some microbes produce chemical shuttles, microbes that can cycle between the cell and a surface. Other microbes need to make direct contact, while other microbes seem to be part of the microbial community for reasons we don't fully understand. The length scales for these wires are becoming larger as we explore this further, because individual wires are something on the order of 10 to 100 nanometers. So, these would be maybe up to microns in terms of connecting cells. Then you have biofilms, which become electrically conductive on the scale of maybe 10 to 50 microns in depth. And recently, there's been a filamentous bacterium which was discovered, which was transferring electrons along a filament on the order of millimeters to tens of millimeters or centimeters. And we don't even know to what extent the current may be transferred in a transverse direction across these biofilms. And a very elegant experiment came out of Derek Lovely's lab where they had a gap between two biofilms, and then they let the biofilm overgrow it, and they could detect current coming from one side across that gap and through the other side. So what microbes are doing this? And to really address that, we said, well, let's go and get the samples we usually get from we use the Penn State wastewater treatment plant, best source we could think of. There's another treatment plant in town. We got some microbes from there to see how things might vary between treatment plants. And then we went out to an anaerobic bog, which would be a source of good iron-reducing bacteria. And we analyzed what grew we analyzed the performance of the reactor over time, and we analyzed what grew in there in 4 different ways. I'm only going to talk about 2 of these. So first, the performance. This is a cycle number. This is feeding the reactor. It runs out of food. We give it more food. It runs out of food. Each one of those is a cycle. You see the 2 wastewater treatment plants had a time here, and over a few cycles they built up. But the bog shot up, and it was much higher, but it was very stable after that. And then over time, the wastewater treatment plants caught up. So as the biofilm acclimated to the most suitable microbes in the system, they eventually converged to produce the same amount of power. What kind of microbes were in there? Well, this is some pyrosequencing showing that most of these samples were all basically delta proteobacteria, these blue bars. There's some other ones, but they're predominantly these. And if you analyze the delta proteobacteria, they're almost all microbes with sequences most similar to Geobacter sulforeducens. So a lot of work to not find out a whole lot other than Geobacter sulforeducens seems to win. And also we used DGGE to show that the bog community profiles were much different than the wastewater samples. The wastewaters generally looked like similar communities. The bog samples were quite different, but at the end, they all look pretty much the same. So that's what we got from 2 different messages. So yeah, I want to have high power, you got to work with Geobacter sulforeducens or like species. Now what about the electrotrophs? These are microbes that could accept an electron from an electrode and then send it on to something else. Dissolved oxygen has been shown, nitrate, but let me talk about the CO2 fixation by electromethanogenesis. The classic theory of methane generation by hydrogenic trophic methanogenesis is that a fermentative organism breaks down, say, glucose, releases some hydrogen and some acetic acid. That acetic acid cannot be used to make methane, but hydrogen can be used to make methane. That hydrogen, those electrons actually go through this cell very fast. It's a very efficient process, And the same is true of the methanogen. They take hydrogen and very quickly convert that to methane. These are very efficient systems. It's almost like, though, that two superhighways are connected by a dirt road, because now you're relying upon diffusion of this hydrogen gas, which is sparingly soluble, to go from one microbe to the other. So why should nature develop like this? It doesn't make sense. So what if there's direct electron transfer between these two microorganisms? Wouldn't that be more effective? Cut the diffusion step out. And that's what we call electromethanogenesis, and we probed that by doing some different experiments, but the first evidence we saw was that we found in nature that fermentative organisms were connected to methanogens by these appendages, and it turned out these appendages were electrically conductive. Doesn't mean that that's what's going on, but that's interesting. And so we enriched cultures using a potentiostat on a cathode. And over time, we got a pretty relatively pure culture, and it was making a lot of methane. And if you look at the amount of methane that you could make based on the hydrogen gas that would come off that electrode, that's how much you'd get. This is how much methane we were getting. So the only way that that could have occurred is if the microbes were directly taking those electrons off the electrode, that they did not have to go through hydrogen evolution. We also showed this with the pure culture, although the effect was quite less compared to the strain or the predominant biofilm from the natural culture. So we're really interested in what makes this all tick. I don't have time to say much more except that, you know, it's really fascinating. You know, who is making the wire? Who's making the plug? Is it you know, that these electrogens make the wire, and then they got a plug and they're looking around for the right socket? How specific is this? Because if you go to the UK, that plug doesn't look that way. And if you go to Germany, it looks like that. So, you know, this wouldn't allow these interactions to occur, but maybe the microbes have figured that out, and they have universal adapters for these different wires, or maybe there's just universal donors, universal acceptors. We don't know how specific this situation is, but we also want to see if we can improve on nature. Can we make artificial bio-nanowires or nano-bio-wires, or can we now engineer this into microbes? And this isn't my work. This is some work out of Guy Bazin at UC Santa Barbara. And he studied the use of these conjugated oligoelectrolytes, which are molecules that can transfer electrons. And they self-insert themselves into the lipid membranes of microorganisms. And that's really convenient because it means it's really easy. But what's really even more fascinating is that when they get inserted into the cells will fluoresce. So you can actually tell that the wires have gone into the cell because they start fluorescing. And what he showed was that a microbe like E. coli, which really doesn't produce very much current, and it only does that indirectly by some flavin-like compounds. When he put these in the E. coli, he could make a lot more power in these microbial fuel cells. So this is with the COEs inserted, and this was with the E. coli by themselves. Not as much power as Geobacter sulfur-reducing-like microbes, but still, he showed it could be done. So that's the microbiology side. What about the engineering side? Can we scale these things up? So in order to do this, you got to come up with the right designs, and you have to use materials which are not expensive, because you're going to need a lot of them. So we've built a lot of reactors at Penn State. They're square, they're cube, they're flat, they're wide, and so forth. And over time, we realized that the more important aspect was what the materials cost, that you could accommodate different architectures, but it was really the cost of the materials. And you have to do this for about $100 or 100 euros per square meter of electrodes. So that's our goal. So if we make a microbial fuel cell 8-10 years ago when we started this, what would it cost per square meter of electrode? Well, you need a tank. You always need a tank. I'm not going to worry about the cost of the tank, okay? I'm just going to worry about the cost of the electrodes. You fill it with wastewater, that's your source of fuel, that's free, cheap. Then you put in your anode, that's where the microorganisms grow on. They're available, they don't cost anything. You have a cathode. The cathode is where oxygen's going to be reduced. You want to use oxygen in air. You don't want to have to bubble oxygen in water because that takes a lot of energy. So if you want to use that, you have to allow that cathode to be exposed to air, but you don't want water to leak out through the cathode, so you need some sort of diffusion layer, like a Gore-Tex jacket coating on the outside, keep the water in. You want to move that anode as close as possible to the cathode because that reduces internal resistance, just like any battery or fuel cell. And if they get too close, they'll short, so you put a separator in there to prevent them from touching. And if you look at this, the most expensive part of this whole operation is really the cathode. It's made of carbon cloth. It's got a catalyst like platinum on it, precious metal, expensive. And it's got a binder like Nafion, which is almost as expensive as the platinum. You add it all together for a square meter, and you come up with the $2,000 per square meter. It's too expensive. It can't be done. Proof. So you spend another few years working on it, and eventually you get down to components that cost about $40 a square meter. So I'm just going to real quickly tell you some of the highlights of how we did that. The first is we needed a high surface area, inexpensive anode. Oh, I'm sorry, no, I forgot I was going to. The first is I'm going to show you how we're going to compose the architecture, and then I'll show you the material. So if we take this little cube reactor like we've been building in the lab, and you put them together, you can create a little airflow pocket between these cathodes. So you want to try and have airflow cells alternating with wastewater or liquid flow cells. So the wastewater would be flowing through there, and the air would be flowing through there. And then, if you pack those together, the width of that cell will tell you how many square meters you can put per cubic meter. So typically, we have systems that run about 20 to 50 square meters per cubic meter. So you can put a lot of these, a lot of surface area in a cubic meter. So now let me tell you how we got the costs down. The first was this high surface area anode. How do you come up with these ideas? Well, you're down in the lab cleaning test tubes with bottle brushes, and one day you hold it up to the light and you realize if you make this out of graphite fibers, then these are electrically conductive, and this is the current collector, and you have a huge surface area for the bacteria to grow on, and the size of the fibers is well matched to those of the bacteria, about 5 or 10 microns versus a few microns for the bacteria. So your design of these chambers now is only slightly modified. You just have a series of brushes as an anode with the wastewater going through in the air channels. And so you start to build reactors like this, and now you can start to, in a very applied way, answer questions you really can't answer very easily. Otherwise, do we use a few big brushes, a lot of small brushes? And so you build these reactors, you send the wastewater through, and eventually, you come up with this sort of situation. You have a big brush here and a cathode, and you want to keep that distance short, so you move that big brush right up near the edge. And if you use smaller brushes and you keep them the same center line distance, you can see this distance increases. Makes sense. Turns out that this brush doesn't work as well as that one. Well, is that because they got farther apart, or because the little brush didn't work so well? Well, you move the little brush closer, and then you find that, yes, when you move this little brush closer, it works better. And it works better because the bacteria here all need to be closer to that surface, whereas here you have lots of areas that basically aren't doing anything. So you want to keep these brushes small and close to the surface. The next is the cathode. Platinum and Nafion, too expensive. So I'm at a conference in Europe, and these guys from Belgium come and say, hey, we've got these cathodes. We've made them out of activated carbon. Yeah. They'll work just as well as platinum. No way. So we try them, and they almost work as well as platinum. And so here's the activated carbon, and here's the platinum on carbon cloth. Activated carbon it costs pennies per pound. It's actually a pretty good oxygen reduction catalyst at the current densities that we are interested in. So we've done some work. Environmental engineers love to work with carbon, so we looked at different types of carbon, and it turns out understanding why it works so well is not so easy. So we looked at some things, and indeed we could see quite big differences depending on the precursor material for those carbons. And now we're trying to figure out which ones will work best, but we're also trying to modify those chemically for improved catalytic activity. And it turns out that binding a little bit of iron into the system worked pretty well. So these are all a bunch of new cathodes with some different treatments. Here's platinum right up here in these orange circles. And here are the other carbons with some different treatments. Then you wait a month, you keep running this thing month after month, and then here's after almost 5 months, and what do you see? You see the platinum ones actually don't work very well anymore. But the activated carbon ones, they're working pretty well. Their performance has only dropped just slightly over that time. Now we know we lose performance over time, and so there's still work to be done in extending that performance, but there's a lot of good fun ahead in terms of perfecting that carbon. So those are the main components. Let's talk about the engineering, which everybody always thinks, well, that's trivial, you just make it bigger. Here's one of the first larger scale MFCs. This was done by a group in Australia at a brewery, Foster's Brewery, probably heard of it. It worked out pretty well, but they actually never published any results on it. And what's really good about that is because they had a website where this picture appeared, and I used that picture in a paper I wrote, and now everybody cites me for their work. It's perfect. This is another one which is being developed in China. The idea is you make one of these little modules, you can make it 4 of these together and get good hydraulic flow, and then you can stack these up. And this system, if this module was used as done here, would be about 10 cubic meters, or 10,000 liters, about 10 times the scale of the other reactor. With microbial electrolysis cells, these are the reactors where I said you have to add in a little bit of power, but you get hydrogen coming off the electrode. We've been able to build bigger reactors a little bit more easily because you don't need that air interface in the system. And so this was one of our earlier reactors. It's about the size of a shoebox, and a very well-wired shoebox. And it had a bunch of these graphite fiber brush electrodes, and we used stainless steel as a cathode. It turns out stainless steel for hydrogen evolution works not too badly. And then this is a 1,000-liter reactor that we built with 24 modules of these brush stainless steel pairs for a total of 144 individual pairs of electrodes. So you're going to go test something like this. You want to do some, get some data before you run out and plop it down. So we got some wastewater samples. We brought them back to the lab. We did some tests, see how well they worked as MFCs, but also as MECs. And as MECs, this is your number you're going for. You're going for a high gas production rate per volume of reactor with that wastewater, right? So if you are a good scientist or engineer, you can see quite clearly we should go to the potato chip factory and do our tests there. But we're not very good engineers or scientists, but we do like to drink wine. And so we went to the winery, which wasn't a bad, it was. But if you're going to go to a winery and you live in the state of Pennsylvania, so if you're going to go to a winery, go to Napa, California, and there you will have a good experiment, or at least a good experience. So we went to Napa, we built the reactor, we went to Napa. This is the current that we expected to get through each of these modules. That was our goal. This is what we got. You could see we did not achieve our goal at most of these, but we didn't do too badly. But when all said was done, we got 16 times more energy in the gas that we got out of the system than the electrical energy we put in. And that's pretty good, except for the one minor detail that the gas we got out was mostly methane, because the methanogens ate up all that lovely hydrogen gas and made methane. Now, there's a group in Newcastle which has done some work with a cassette design which allows them to collect that gas more efficiently. Although they didn't break even on energy, they only recovered about 70% of that energy in the hydrogen gas. Okay, so there are other technologies that you can bridge. Now that you know that you can use microbes to make an electrical current or to accept an electrical current, you can do a lot of different things. For example, you can desalinate water without electrical grid energy. you can recover phosphorus on these cathodes by changing the pH in certain waste streams. The one I want to spend the last little bit of time talking about is the salinity gradient energy and waste heat energy. So again, just to remind you, there's maybe a terawatt here and another terawatt here in waste heat and salinity gradient energy that's out there to be used if we can figure out how to do it. So I like to say, you see a lovely ocean. You probably have other thoughts than the one I'm telling you about, but if you think about the ocean and water running into that ocean, and you translate that to water going over a dam, there's as much energy to be recovered here as the energy recovered from water going over a dam 270 meters tall. And that's why you can generate so much energy from a salinity gradient difference. It makes sense. It takes a lot of energy to squeeze fresh water out of salt water. So thermodynamically, everything's reversible. If you have those fresh and salt water solutions, there must be a lot of entropic energy to be recovered. So there are three basic technologies: Pro, Red, and what we call cap mix. There's another variation, which is a battery-like technology, which we like to call bat mix because we think it sounds cool, but nobody else calls it that. Pressure retarded osmosis, Manny Elmelech talked about, so I'll just tell it very quickly, and then I'll spend most of my the time remaining about these last two technologies. Now, when I initially looked at these technologies, I didn't just start off looking at the specific technology. I also thought about trying to incorporate these technologies into microbial fuel cells. So you have three possibilities: MFCs and pressure retarded osmosis, MFCs in red, MFCs and cap mix. Now this, to anybody who has any business acumen, seems to be the most lame idea ever. Because what you're doing is talking about taking one technology which hasn't worked and combining it with another technology which still isn't commercialized. And you ought to get nothing out of that. So here's my analogy. You have a block of wood, and you want to split that block of wood, right? So somebody gives you a stick. You whack at that block of wood. You don't split it. So you say, well, forget that. How about if instead I use a different technology? I'm going to use a sharp piece of metal. That doesn't work very well either, does it? But you can see where I'm going here. If you take these two things and you put them together, you get a technology that actually works pretty well. So the idea is the technologies are combined because they overcome the limitations that they currently suffer from. Synergistically, they work better. So let's see what happens with pressure retarded osmosis. Pressure retarded osmosis, you have a low salt solution, a high salt solution, and the water wants to run from the water wants to go in and dilute out the seawater. And that will cause the seawater to be pressurized. You release that pressure through a turbine, and you can make electricity. So that's pressure retarded osmosis. How would you combine it with a microbial fuel cell? Well, wastewater is essentially river water. It's very low salt. You put a membrane here that allows the water to pass, but not the ions. You dilute out this. You pressurize the water, so you have your microbial fuel cell making electricity. You can even make your air cathode over here, so you also carry protons through that membrane. That helps produce power. And voila, you could make electricity from the pressure as well as the MFC. But it turns out it's pretty complicated, and this is probably a good way to desalinate water. It's probably more useful for that reason than for making power. So that's not such a good idea. What about reverse electrodialysis? Reverse electrodialysis is the reverse of electrodialysis. So what is electrodialysis? Electrodialysis is how you can desalinate water. You put seawater in all these chambers. These are anion exchange membranes and cation exchange membranes. And then you apply a potential across all these membranes. And what happens is all the negative charge wants to go to the anode, and all the positive charge wants to go to the cathode. But they can't get there. This negative charge goes that far, and that's it. It stops. Can't go through the cation exchange membrane. So you end up diluting this one out and concentrating this one and so forth for the numbers. So that's electrodialysis. It takes energy and a lot of it. Reverse electrodialysis is the opposite. You start out with river water and seawater, and now you put these ion selective membranes here it only allows the negative charge to go that way. It only allows the positive charge to go that way. That creates electrochemical potentials of about 0.1 to 0.2 volts per membrane pair. And then you string a bunch of these together, and you can make electrical current. Just like you string a bunch of batteries together, you increase the voltage. As you string these together, you increase the voltage as well. And if you have enough of these membrane pairs, you can split water on an anode and produce hydrogen on the cathode. So you need 10 to 20 membrane pairs just to get going. That just gets the reactions going at the electrode. So you need a lot of these to do anything useful. But I thought, wait a minute, you know, in a microbial fuel cell, I already have thermodynamically favorable reactions. So I took a stack like this, and I inserted that stack into the microbial fuel cell. So now what you have is you've got this same setup, but you have an anode which is producing electrical current. You're not forcing a reaction there. You've got an air cathode where oxygen is being reduced. You're not forcing the reaction. In the middle, you've got this saltwater battery. So you put them together. It looks something like this. We actually had serial flow going through these membranes. But when we put that in there, we thought, well, we're not really sure this is going to work, but it worked better than we could have believed, because now, instead of producing about 0.5 volt, we were producing more like 1.5 volts. The power that we could get from these individual systems, essentially none. This is just what a red stack will produce when there's no fuel left in the system. And this is what the MFC can produce by itself. You don't add these up and get this. So together, they actually work better than they did individually. And the energy efficiency was 42% based on energy in the acetate, which was the fuel here, and the salinity gradient energy entering and leaving. Now, since we now have a built-in power source, we don't need an external power source anymore to provide that extra voltage for doing hydrogen evolution at the cathode. So this is, I won't say it's a perpetual motion machine, but if you feed the anode and you keep pumping fresh and salt water through here, you can sustainably make hydrogen with no net carbon footprint. Okay, this brings me to the last thing, and I think I'm doing pretty good. Right? So this will take about 5 minutes. And I leave this for the end because you've been sitting there for 50 minutes, 45 minutes, and your brain's starting to get a little tired. And now I'm going to make you think really hard. Okay, so there's the third way to make electricity, is what we call this cap mix process. And there are at least 3 different ways to do this. One that involves a ion exchange coating, one that doesn't, and one which is, like I said, uses like these battery type of reactions. I'm just going to explain one of these, and I'm going to explain it from the perspective of just having sodium and chloride ions. So what we're going to do is we're going to alternately expose these electrodes to fresh water and salt water. So that's the idea. And we're going to form a cycle. So I'm going to have cycle 1, cycle 2, 3, and 4. Okay? And I'm going to start with cycle 1. This is this stage right here. And what I'm doing is I've got salt water in this, and here's my electrode. And electrons are flowing as I put that salt water in. This is the start of the cycle. You can see there's current here. Those electrons are flowing. They're not flowing naturally. They're flowing it because I'm putting charge into that system. I hooked it up to a little power source, and I'm making that current flow. So here you go. Voltage is going up. Exchange, this is more current, more current, more current. Then I stop it here. And then I open the circuit so no current can flow, and I add fresh water. And when I add that fresh water, these ions that were all drawn up right against the electrode, they expand. Ions expanding do work. And the outcome of that is the cell voltage increases. And now I connect up the circuit again, and I let current flow, and current flows spontaneously now back into here as that charge is released. That's step 3. And then I finish it by opening the circuit again. I put salt water in. That drops the voltage. And then I go back to cycle 1. Okay, so salt water charging, ions towards the surface, opening the circuit, expanding the electrical double layer, connecting the circuit, allowing current to flow, opening the circuit, adding salt water, and I form this cycle. And the energy, net energy, this is everything below this line is the energy you put in, essentially, not quite correctly, but you get the idea that this is your voltage charge window, and that's the net energy you get out of the system. So it's kind of like a Carnot cycle. But this is driven by fresh and salt water energies. And so this is the energy window you get from what I've described. This is the energy window you get when you put these capacitive electrode systems into a microbial fuel cell, and I'll show you that in a second. And this is what happens if you additionally you charge the current past the point at which we would say it would zero out at. And I won't go into that further, but you can see that the voltage windows get bigger. So just comparing this one to that one, what we found was that when we put these capacitive electrodes in this ionic field, that we could switch these solutions out and extract somewhere between, we could extract 65 times more electrical power out of that system than previously been accomplished with that system. And we got 46 times more power production. Now these power densities, about watt per square meter, they're not high. The power densities for microbial fuel cells are not high, but these reactors are typically quite large. And so, you know, wastewater treatment plants operate very well with this technology, even though it's quite large. So that's how you make energy from salinity gradients. And if you saw Manny Elamelic's talk, I'm sure he mentioned waste heat energy and thermolytic solutions. But the whole idea of capturing waste heat energy is to create salty solutions using that waste heat. So what you do is you use something like ammonium bicarbonate. And ammonium bicarbonate has a very unique property that if you heat it just slightly, 45, 50, 60 degrees C, the ammonium and carbonate will distill off. So you can capture that and condense it down into a high concentrate ammonium salt solution, and you're left with a low concentration solution. So this is your river water now and your seawater. And then you use them and you end up with something in between, and then you redistill it back to freshwater and seawater. So you create this cycle, a closed loop cycle of regenerating these salt solutions. And if freshwater seawater is 270 meters, this can be something more like 370 meters, and then there are other combinations of things that you could use to generate even more, or perhaps less as well. So just to say what happened when we used ammonium bicarbonate into this system, we used wastewater now, no acetate or anything. We just took domestic wastewater from a treatment plant, and this is how much power we made in the microbial fuel cell by itself, and this is how much power we made with that wastewater and this battery in here. What we found was that the battery pushed the system along. The main reason hydrogen fuel cell works so well is the Nafion membrane. The main reason a microbial fuel cell works so poorly is that there are many things that can exchange charge, not just protons. But the potential of driving ionic flow with this ammonium bicarbonate battery flattened out these potentials. And what it means that when these flatten out is that as current goes up, you have higher and higher current and the same voltage. So your power goes up. Power is current times voltage, right? So you can make more power. And not only did we make more power, about 10 times more, which was totally unexpected, but we also treated the wastewater faster because we figured out that if you're going to take that organic matter out faster, you're making more power, you're making more current, you should do it in less time. And indeed, we did it in a matter of a couple hours, where typically the microbial fuel cell would have taken a day to do the same thing. So that's it. You know, I don't know how to make conclusions about all that, so I just threw in a few little tidbits. One, walk out of the room. What do you remember? Some microbes make electricity, some accept it. Two, you can make these different platform systems to accomplish a lot of different things. You can make electrical power. You can drive carbon neutral fuel production in the form of hydrogen or methane. You can desalinate water. You didn't see that one, and do many other things. Virtually anything you know how to do with electricity. Yes, they are being built at larger scale, but by no means is that sufficient. That's only a few things around the whole world of activity. That's not enough work being done at the pilot scale stage. It's not trivial engineering. We need to do more of it, and the more we do of it, the better we'll get of it. So stay tuned, I guess. I want to acknowledge my research group, who, as we all know, they do all the work. This is my current group at Penn State. And I'd also, just because of this particular year, I was on sabbatical the whole fall at Ghent University, supported by the Franck Foundation. And but I also work with a number of other collaborators at a number of other universities, and I wanted to acknowledge them as well, because they certainly have contributed to the work that I've shown you here today. And with that, I'll end my talk, and if we have time for questions, I'd be happy to answer any you might have. And there are some connecting points: YouTube, Twitter. I don't tweet that much. But if you're ever bored, you just go to the webcam and watch that fan spin. Your afternoon will breeze by because you'll probably fall asleep. But anyway, thank you very much.
We have time for a few questions before our reception.
One of the problems with fuel cells is that historically they've degraded in time.
Yes.
And you alluded to that, showing something after 40 hours. Do you have any, what happens after a year or 2 years, whatever?
Yeah, that's a good question. So, you know, think of the fuel cell as being the anode and the cathode. The anode is what Mother Nature devised, microbes that are very efficient at oxidizing organic matter and getting them to electrode. They don't foul that surface. In fact, they tend to grow, the new ones grow at the surface, and they push the old ones away. A year later, spot on, no change in performance whatsoever. The cathode, we designed that. A year later, not so good. Ours showed about a 15 to 25% drop in power after 1 year. So it's really the cathode that we have to work on. And one of the nice things about some of this cathode work we've been doing is we think we're starting to see ways to protect that cathode. And it's, you know, the question is, what is the catalytic activity being lost? Is it being just fouled? Is it just stuff getting to that surface? Can you regenerate it? So we're starting to answer some of those questions, but that will be eventually, because you want these things not to run 6 months or a year, you want them to run many years. And so that, in the end, will be the key. And I think it's reasonable that we could do this for several years, but we will have to clean and regenerate them just like we do membranes and these other processes.
Biofilms on the anode and cathode get attacked by any other organisms that are in the wastewater?
You know, I have a PhD student who'd really like to be able to answer that question. He hypothesized that there were viral infections that would set in on these anodes because they become so predominantly one type of microbe that eventually it must crash. And we had this MFC cam a couple years ago. We had it crashing very regularly, so we devised an experiment to see if viruses could be causing that. And ever since we posed that hypothesis, it's never crashed. You know, and from that perspective, no, there are always grazers, things that are living in the system, preying upon the biofilm. That goes on. We don't know nearly to what extent that might be important. It doesn't seem to be very important in activated sludge wastewater treatment plants, so by analogy, we might say maybe not so important. But that's a question that we haven't really gotten to. Right now, we mostly worry that we get these thick, foul biofilms on the cathode, and that's a greater concern.
I'm just curious if you could compare the power generation from the wastewater to sort of your typical, very simple methane generation when you do anaerobic digestion.
Yeah, actually, I have a good slide on that. Don't have it at hand. So if you look at the MFC with the red stack made of ammonium bicarbonate, it approaches the power densities of anaerobic digesters. So that tells you that without that, they're like an order of magnitude or more, less. The organic matter concentrations in an anaerobic digester are very high. And the organic matter concentrations in microbial fuel cells are quite low. So that's one of the problems. But the other thing is just that everything in the volume in an anaerobic digester is producing methane, whereas in the MFC, it's only that biofilm on the surface. So you got to cram a lot of area per volume in to start to boost it up.
So, going to the future with these technologies, do you envision that they would be applied at very large scales, or for example, for a whole wastewater treatment plant, or do you think that the economy of scale would still allow? I mean, there will not be an economy of scale, and they would still be applicable at, a few fuel cells at a much smaller scale. Which leads me to my second question. For example,
Well, I might forget the first one first.
Okay.
So let me answer the first one. So yes, there's probably an economy of scale. It's just, and right now we're faced with a dilemma. You make one car, it costs you $1 million. You make 20 million cars, it costs a lot less per car. Okay, so there's that sort of scale issue. But intrinsically, we build these things this big, and then we build them bigger, and they scale in. There's not some great failure of that scale, but we know we can't get them too big because electrical resistances go up. So we're going to make a lot of smaller ones, but we do believe we can replace a wastewater treatment. We can take that tank, which is now being bubbled with air, take away those aerators, drop in all these stacks of microbial fuel cells, and accomplish the same thing in that same space. Probably with some add-on treatment, but that's another subject.
So you think you need the infrastructure of a wastewater treatment plant for these? So you don't envision, for example, that you could have them at a building scale?
Okay, so let's say I had one for my home. I would power the front door light.
Okay.
So you don't get that much power from it. However, if I had some grass clippings or fermented material that I could bleed into there, now I could start to produce some useful power. And so we do envision that this is, for example, a good way for sanitation in the developing world, as well as power generation in the developing world, because we can make very small reactors, and they'll weigh a lot, so nobody will carry them off and resell them somewhere else. They're just not practical. So we do think you can do it for a household, you can do it for community, you can do it for a city.
So my second question was, I mean, if you have wastewater that's also hot, because typically when wastewater is generated, first it's hotter. Can you combine the heat and the wastewater ones into some combination that will?
I can tell you the MFC will work under higher mesophilic, thermophilic conditions. But if you have ammonium bicarbonate and the water's 60 degrees, you're never going to keep it in solution, so that wouldn't work. But you can use sodium chloride. If you are working at those temperatures, you can distill off the water and keep the sodium chloride and still get the same low concentrate, high concentrate solution. So maybe that might work, but I don't know. The economics would probably be.
So a problem that occurs with like the Penn fuel cells and others is that you have major mass transport limitations getting reactants to and from the catalytic sites. You've got to have some major mass transport limitations getting your organic matter in and out of the cells, but I didn't hear any consideration of how those mass transport limitations are affecting anything on power density.
So do you speak fuel cells and electrochemical impedance spectroscopy?
Somewhat, yeah.
Okay, so you can use EIS to determine where your resistances are and whether they're charge transfer or mass transfer or ohmic resistances. And right now, we think a lot of the resistances are in the diffusion of molecules to the surface. And we think most of those resistances are protons getting to the cathode surface. Because when I release a proton over here at the anode, and I consume a proton over here, charge balances, but it probably balances because a sodium or a potassium or a bicarbonate ion balance the charge. And so the problem we face is charge is being balanced but not by protons. And so we have very big limitations on probably proton transport, high pH at the cathode, which is our main problem.
Despite the fact that you have a naphthalene membrane?
No, in these single chamber systems, we try not to have that membrane. It's way too expensive. And if we had that membrane, actually you know how you have these papers that you say, this has been cited so many times. One of my most highly cited papers was, we took the PEM membrane out and we made more power. It's like, well, that seems, But you think about it, every fuel cell up until that point had a PEM membrane. And one day we realized we didn't need it because the water was the proton carrier. And so the water is like the dirt road again. The Nafion is a super well-developed material, but Nafion doesn't work once you have other ions other than protons, because the other ions just link up with all the sulfonated groups, and that's it. There's no more protons being transferred in it. So we try and avoid the membranes. Of course, unless I make a red stack, and then I have to write another paper which says we need to put many more membranes in the microbial fuel cell, as opposed to the first one where I said don't use any membranes, right? Yep.
Is the yield biomass per COD removed in your fuel cell less than in a conventional treatment plant?
That's correct. So in an aerobic process, the rule of thumb is you get about a half a gram of volatile solids per gram of COD removed, or something like this. In an anaerobic digester, it's like a 1/10 to 100th of that, right? This is more like an anaerobic digester because, number one, it's an anaerobic process, and number two, we're getting a lot of energy out of this, right? The anode potential is right up against the half-cell potential for acetate oxidation. So the microbes are getting like this much, and we're trying to get this much, but we're only getting this much because that much is going to waste heat and, waste inefficiencies. So the microbes get this, therefore they can't make much biomass. It works to our advantage, and in fact, we anticipate that you could reduce the operating costs of a treatment plant, by a factor of 5, not a factor of 5, but half the energy at a wastewater treatment plant goes into aeration typically, but about half the expense is going to sludge handling. So if you cut the sludge handling down by a factor of 5 or 10, you can really diminish those sludge costs.
So I'm going to ask the last question.
Yeah, okay.
So do you have to get the proton across? I mean, because you have an open system, because you're flowing through, you can have another counter reaction, period. So it seems like that would be...
Yeah, you don't, one of the great fallacies of that cartoon is it shows you the proton moving through. But it's exactly what you're saying. You produce protons over here, and you consume them over here, and those protons may never get over there.
Right, but couldn't you... you would be increasing the pH of the wastewater slightly.
So the wastewater pH goes down, more protons at the anode. The pH of the wastewater goes up because all the protons are consumed.
But for example, if in my work we deal with potassium, and you have potassium near oxygen and a little bit of carbon, and you get these carbonates that pop out that end up being hell on the cathode. But if you can flush them out, you have carbonate in the solution that actually tends to neutralize things. So if you have other positive ions in the solution...
If you have a wastewater with a good high alkalinity, that really helps you. And that's exactly right. The carbonate buffering really helps you.
Okay,
Yep.
cool. Well, we have a reception outside, so I invite everyone to join us there. And let's thank Dr. Logan again.
Thank you.